Very good morning, dear students. The next topic in unit number 2, the structure of protein. This is the most important topic in this particular unit, as we have been discussing the basic fundamental aspects of amino acids and proteins, and we have seen that proteins are the macromolecules of life. They have a lot of functions in the body, and each and every part of the body is made up of proteins. And I have already mentioned, in order to perform the function, the ultimate reason is the structure. So what is the structural features of protein? That is what we are going to discuss in this particular class. Generally, proteins have 4 characteristic structural conformation: primary, secondary and supersecondary, tertiary and quaternary structures. And in this particular class, we are going to discuss primary and secondary structures of the protein in detail. Please note that this is very important aspects in biochemistry, and we are going to discuss the core structural biology aspects of primary and secondary structure of proteins. And before we are going to discuss, we will discuss briefly about the protein structure. So as you all know, protein structure is nothing but it is the 3-dimensional arrangement of atom in an amino acid chain that constitute the polypeptides. The proteins are long chain polymers, and that polymers are polypeptides. So that formed from the sequence of amino acid which are connected by peptide linkage. A single amino acid monomer is known as a residue, which indicate a repeating unit of the polymer. And the protein form by amino acids undergoing the condensation reaction, in which the amino acid lose one water molecule per reaction in order to attach to one another with the peptide bond. We will see. Okay, so 2 amino acids are combined to form a peptide molecule with the condensation reaction and removal of water. And in order to perform the biological function, the protein need to fall into one or more specific spatial conformation. Okay, that driven by the lot of bond, lot of non-covalent interactions such as hydrogen bonding, ionic interactions, van der Waals interactions and hydrophobic interactions. So all these are small covalent bond that has got a lot of relevance, and these covalent bonds, non-covalent bonds are determine the stability of protein structure. So in order to understand the function of the protein at a detailed molecular level, it is very essential to study the structure of the protein, and it is of course important to determine the 3-dimensional structure of the protein. And we can use the technique as we have discussed previously: X-ray crystallography, nuclear magnetic resonance spectroscopy, cryo-electron microscopy, the latest techniques. Okay, in 2017 scientific community got Nobel Prize, cryo-EM, and dual polarization interferometry are some of the techniques that can be used to determine the structure of the proteins. Now I told you the most important stabilizing factor of a polypeptide is peptide bond. Okay, so peptide bonds are the bonds that connect the long polypeptide chains. So the amino acid units are linked together to the carboxylic acid group and amino group to produce the primary structure of the protein. And there is a strong covalent bond that is known as peptide bond which is a chemical bond formed between two molecules when the carboxylic acid group of one amino acid is interacted with the amino terminal end of the other amino acid, thereby the release of water molecules. That is what I told you, hydrolytic condensation. And depending on the number of amino acid molecule composed in a protein chain, the peptide can be dipeptide, tripeptide, oligopeptide, polypeptide and macropeptide. So these are the classification that depending upon the number of amino acid. So dipeptides are if the peptide consisting of only two amino acid and one peptide linkage. And tripeptides are the amino acid, the protein peptide consisting of 3 amino acid unit and 2 peptide residues. And oligopeptides are the peptides which contains more than 10 amino acid residues. I told you less than 30 and more than 10 amino acid residues. Polypeptides are the protein with a larger number of amino acid residues. And macropeptides are the protein consisting of more than 100 plus amino acid. Okay, so these are the overview of peptide bond. So this is the formation of peptide bond. I told you this is one amino acid. This is written in the form of sitter ion. It has got an amino terminal end and a carboxyl terminal end and a hydrogen and a side chain R. Okay, so here the second amino acid, okay, it has got an amino terminal end, the side chain that is denoted as R2, and this is hydrogen and this is the carboxylic acid group. So what is happening? The carboxylic group of one amino acid interacted with the amino terminal end of the another amino acid. Here you can see NH2 or NH3. Here you can see hydroxyl group. Okay, if my negative is not there, then hydroxyl group. Then what will happen? This will be produced 2 hydrogen from here and 1 oxygen from here. It will go through a water molecule and you can see, and both the molecule will fuse. Okay, NH3 is written like this, R1 it is here, C it is here and H is here. The C double bond O is here, and you can see that this O is gone. Okay, and what you can see, then it is bonded with this one. H2 is gone, then only H is present, then that is NH. Okay, then this C R2H, then COOH. This is the carboxy terminal end and this is the amino terminal end, and this CONH is known as peptide linkage. This is a rigid, very strong covalent bond. Okay, and this is known as a peptide. This is known as a dipeptide. This is a single peptide unit. Then what will happen? The next amino acid will come here, and this end and the next amino acid will be interacted to release the water molecule, and you will be having the next amino acid, and so on. It will add n number of amino acid and form polypeptide chain. Okay, then what are the characteristic features of this peptide bond? And this peptide bond is a rigid and a covalent bond which is formed due to the hydrolytic condensation reaction. The first property is rigid and covalent. The second feature is it is formed due to the hydrolytic condensation between amino group of one amino acid and carboxyl terminal group of the other amino acid. And the peptide is, this bond is a planar. That means you say the C, H, O and N, these 4 residues are oriented in a single plane. And this bond has got a partial double bond characteristics. Okay, always there is a shared form of electron, from it will migrate from here and like that partial double bond characteristics will be here. This will be coming here. Okay, and this will be going here. Okay, this will come here, this will go back, this will come here, this will go back. Okay, so like that there's a partial double bond characteristic due to the resonance stabilization. And it can exist various isomeric forms such as the cis and the trans form. Okay, the trans varieties are more stable than the cis variety. And the most important silent feature of a peptide bond is it has got a 3 dihedral angle or torsion angle, that is known as phi, psi and omega. Okay, and 2 bonds are rotatable and 1 bond is non-rotatable. And what is phi? Okay, we will see that the degree of rotation between C alpha and C in a peptide unit is known as psi, and the bond length is 1.51 angstrom, and this is a rotatable bond. So the bond between C alpha and C is known as psi, and that is the bond length is 1.51. Then the degree of rotation between N and C alpha is known as a phi, and the bond length is 1.46 angstrom, and that is also rotatable. Then the angle between C and N is known as omega, and the bond length is 1.32 angstrom, and this is non-rotatable. Okay, so these are the major types of torsion angle, rotatory angle in the polypeptide chain. And this is what I told you. Here you can see this is C alpha and C and C alpha. Okay, simply you can see the bond between the C alpha and N, that is known as phi, and the bond between C and C alpha is known as psi. These two are rotatable bond, and it is non-rotatable bond, C and N, and this is CONH. This is known as a peptide bond. Okay, here you can see the bond length is 1.33, 1.46 and 1.51 as we have discussed, and here the bond angle is between these two, it is 120.5 degree, this is 123.5, and this is 116 degree, and here this angle is 122 degree, and this is 119.5 degree, and this is 118.5 degree, and this is 1111. Okay, and you can see here also the bond length is the C double bond, this bond length is 1.24, this NH is 1. Okay, and this I told you C, H, C alpha and O. O, C alpha, H and C alpha. Okay, and N. Okay, this is involved in a single plane. Thus this particular, this is a this is known as a planar, and I told you it can exist both the direction. If C alpha and this one and NH in the opposite direction, that will be trans, and if it's in the same direction, that will be cis. Okay, but trans direction is very common, that is very stable. So this is the major torsion or dihedral angle is polypeptide chain. This is very, very important. Then we are going to discuss about the protein structural configuration based on the peptide bond and the major polypeptide. Keeping in your mind, the protein has 4 structural organization. Okay, one is known as primary structure, next one is known as secondary structure and super secondary structure, next one is known as tertiary structure, next one is known as quaternary structure. In your syllabus, they have mentioned the first 3: primary structure, secondary structure, tertiary structure. So overview very briefly, primary structure is a linear sequence of amino acid which are connected by peptide linkage. Okay, like this. Okay, then if this primary structure, this is connected by peptide linkage, and this primary structure is form a local conformation by in the form of a zigzag manner or in the form of a helical like fashion, that is due to hydrogen bonding, that is known as secondary structure. Okay, here you can see this secondary structure or a zigzag structure. This is known as secondary structure. This is the primary structure, and this secondary structure completely fully folded to produce a particular motifs or domain is known as the tertiary structure. One single completely polypeptide chain is completely fully folded, and a characteristic 3-dimensional structure is known as a tertiary structure. Here I would like to call it something called as chain or a motifs or domain, and one single motifs will be there, and that there are lot of forces that stabilizes the structure. Here the stabilizing force is peptide linkage, here the stabilizing force is hydrogen bond and peptide linkage, here the stabilizing force is so many weaker interaction as I have shown in the beginning: hydrophobic interaction, water matrix interaction, ionic interaction, etc. This quaternary structure is multiple chains, okay, like this multiple chains are combined to form, that is known as quaternary structure. Okay, this is single chain, tertiary, this is multiple form, okay, quaternary structure. And the importance of this particular things, all the 4 level of structural organization got the prestigious Nobel Prize in physiology or medicine or in chemistry. Okay, so that is very, very important. Now in this particular class we will discuss the primary structure and secondary structure in detail. So this is what I told, the primary structure, linear sequence of amino acid, tyrosine, lysine, alanine. I told you we can represent in 3-letter code or single letter code, lysine, alanine, valine, aspartame, leucine. If you write it like this, this is the primary structure. And this same thing, if it is followed in the form of a zigzag direction, in like something like an asbestos sheet, something like a local structure, that is the beta sheet. And if it is formed in the form of a helical like structure, that is alpha helical structure. And if this beta sheet multiple form produces a single subunit, a single structure is known as tertiary structure. And multiple domains, this is known as a domain. Okay, particular thing is known as a motif. This is a motif. Okay, so motifs are nothing but small structures of amino acid of a polypeptide chain that has got a particular structure and a function, that is known as a motif. Okay, motifs are small structures of amino acid that has got a particular structure and function is known as a motif. Okay, so multiple motifs, that so many motifs combines to form domains or chain. Okay, this is one chain, multiple motif. Okay, so here you can see one chain, multiple chains to form quaternary structure. So this is primary, this is secondary, tertiary, and this is quaternary. Okay, and first one, primary structure of protein. The primary structure of the protein refers to the sequence of amino acid, as I have shown here, which are connected by peptide linkage. Okay, the primary structure is held together by peptide bond that are made during the process of protein synthesis. Okay, how it is produced? As I said, the two end of the polypeptide chain. Okay, so that has got one terminal is amino terminal and next terminal is a carboxy terminal. Here it is an amino terminal and the next one is the carboxy terminal. Okay, the carboxy terminus of one amino acid is interacted with the amino terminus end of the other amino acid. The primary structure of the protein is determined by the gene corresponding to the protein. A specific sequence of a nucleotide in DNA is transcribed into mRNA, which is read by ribosome in the process of translation. We will discuss the molecular biology mechanism of translation in the next semester, molecular biology. Then the sequence of a particular protein is unique to that of the protein and that defines the structure and function of the protein. Okay, unique to that of the protein and that has got a structure and a function. So this is the very, very important aspects. And this is what I told. This is the amino terminal end. You can see glycine, proline, tyrosine, glycine, like that you can see. Okay, so this is the carboxy terminal end. Okay, here you can see first amino acid. Here you can see R1, R2, R1, R2. This R1 is the side chain of first amino acid. R2 is the side chain of second amino acid. Here the CONH link. Then R3, then R4, COH link, then R5, R6. Okay, you can see the trans configuration. Okay, so this is first one bond, second bond, third bond, fourth bond, fifth bond. This all the things are peptide bond. So like that, if the proteins are stabilized, that is known as a primary structure of the protein. And there is a great study conducted by the primary structure of the protein. Okay, the first this particular idea that is proposed by Dr. Frederick Sanger when he was just a very young scientist. Okay, when he was an young scientist. Okay, and he studied about the bovine insulin. So bovine insulin, you know that it's insulin. It has got a two chain A and chain B. Here you can see in chain A there is 21 amino acid, chain B 30 amino acid. From this is amino terminal end to the carboxy terminal end, it is going. From here also amino terminal end and carboxy terminal end is going. Okay, the there is a cysteine and cysteine. The seventh one, 1, 2, 3, 4, 5, 6, seventh one, the seventh cysteine between the first chain and second chain, there is there is a disulfide linkage. Similarly, here also you can see. Okay, then there is a 19th one. Okay, the 19th one, then this is between the 7th one, 1, 2, 3, 4, 5, 6, the 6th one, 7, 8, 9, 10, 11. Okay, 6th and 11th one, there is a disulfide linkage. Okay, and here you can see there is a 90th one, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20th. Okay, the 20th one of this one and the 20th one of the B chain. 20th chain of 20th amino acid in the case of A chain, and the 20th amino acid in the case of B chain, there is a hydrogen, there is a disulfide linkage, not hydrogen bond. This is stabilized by peptide linkage, and this is stabilized by disulfide linkage. This beautiful study is conducted by Dr. Frederick Sanger, and he has received the prestigious Nobel Prize when he was just 30s. Okay, and he is the person, he studied a majority of the concept in primary structure of the protein. So the primary structure of the protein, the concept which comprises of the linear sequence of amino acid that is connected by peptide linkage, is proposed by Dr. Frederick Sanger, and when he was studying about bovine insulin. Okay, and he received the prestigious Nobel Prize, and he was the one among the very small scientists. He is the third scientist. He got Nobel Prize twice. Okay, one is for the study structural elucidation of bovine insulin. Then next one is the second one when he was an 82-year-old. Okay, he got the Nobel Prize for the Sanger sequencing. Now also we have been using that. That you will study in the further later subsequent higher classes you will study. Okay, so this is the greatest contribution of Dr. Frederick Sanger. That is what I told you. This particular study has got the Nobel Prize in the primary structural elucidation of proteins. Then, in connection with the primary structure and structural elucidation, a great contribution made by an Indian scientist called Gopala Samudram Narayanan Ramachandran, or G.N. Ramachandran. He was from Indian Institute of Science, Bangalore. Okay, I got an opportunity to visit his laboratory. Okay, and you know that he has proposed something called as Ramachandran plot. Okay, Ramachandran plot. So this plot will clearly indicate whenever the protein structures is produced, what should be the torsion angle of a polypeptide chain in order to make the secondary structural conformation. What is the possible secondary structural conformation of a polypeptide chain if the polypeptide chain is undergoes one particular xi and phi angle? And the conformation of a main polypeptide chain can be completely determined if the values of phi and psi are for each amino acid in the residue, the chains are known. In a fully stretched polypeptide chain, the phi is equal to psi, that is 180 degree centigrade. So, G.N. Ramachandran in 1963 studied that amino acid residues in a polypeptide chain cannot have just any pair value of the main alpha and beta. They are different value. They are having one value of psi and one value of phi. Okay, the allowed ranges of phi and psi can be predicted and visualized, something called a steric counter diagram called Ramachandran plot. This is otherwise known as steric counter diagram. So what is clearly indicating? So this is a plot, it's a graph by the possible phi angle taken along x-axis and possible phi angle taken along the y-axis. Okay, from 0 to 180 degrees, 0 to 90 plus 90, that is plus, and 0 to 90 to 180, that is minus. Okay, so this torsion angle of each residue in a peptide defined the geometry of its attachment to the adjacent residue, how the proteins are interacting, the amino acid residues are interacting, and how it is producing a stable secondary structure, how it is positioning, where should position in order to create the secondary structure in order to make it planar. Okay, and the peptide bond relative to two adjacent planar peptide bond, thereby torsion angle determine the conformation of the residue and the peptides. By making a Ramachandran plot, protein structural scientists can be determine which torsion angle are permitted and can obtain the insight into the structure of peptide. So this is the greatest contribution made by an Indian scientist called G.N. Ramachandran from Indian Institute of Science, Bangalore. Okay, and he was the founder of molecular biophysics department in Indian Institute of Science. Okay, then the regions. In this particular plot, the regions are classified into two: allowed regions or favored region. Disallowed regions are non-favored region. So this is the greatest scientist, Dr. G.N. Ramachandran. He was a very simple teacher. Okay, so that is the greatest human he was. And this is the plot. Okay, here you can see 0 to minus 180 and 0 to plus 180. This is known as a phi angle, that is a phi, and this is psi. And this particular plot has got a 4 region. Okay, this is divided into 4: upper left side, upper right side, lower left side, lower right side. In the case of y axis, here it is 0 to minus 180 and this is 0 to plus 180. Okay, this is minus, this is plus. Okay, and any residues, amino acid residues, if positions in the upper left side of the Ramachandran plot, their phi angle and psi angle both are comes under this particular residues. These are the most allowed region. This shaded region, this is known as most allowed region. The blue, the red colored region, yellow colored regions are known as additionally allowed regions. So any residues are comes under this particular region, then it is said to be beta sheeted protein. Okay, any residues, okay, the phi and psi angle comes under this particular region, then it will be right-handed alpha helical structure. Okay, any residues are comes under this particular region, then left-handed alpha helical structure. Okay, so here this you can see, this is a yellow color, this you can see yellow color regions. Okay, so this white regions are known as disallowed region. So the protein structure should be stable, then none of the residue should fall into this particular region. Okay, the residue, if they acquire, okay, the torsion angle or dihedral angle in this particular region, it will not produce a stable structure. Okay, the steric clashes, they will be minimized, and this particular region only the steric clashes will be minimized, they will be able to produce the structure. Anyway, you will study details of Ramachandran plot in your higher studies or higher classes. And this is what the possible plot. Okay, so I told you this is anti-parallel beta sheet, we will discuss. This is beta sheet, the psi and phi angle. Okay, this is psi and this is a psi and this is phi. Okay, this is a right-handed alpha helical structure. Okay, this is a left-handed alpha helical structure. So any residues are comes under the upper left side of Ramachandran plot, that can be anti-parallel beta sheet or parallel beta sheet. Okay, or sometimes collagen helix. Then any residues comes under some places. Okay, this is the upper right side of the Ramachandran plot, then it will be left-handed alpha helical structure. Then any residues comes under lower left side of Ramachandran plot, right-handed alpha helical structure. This green region, like this is same plot, okay, in a different style. So this is a most favored region or allowed region, and these regions are known as additionally allowed region, and this is known as disallowed region. So this is the greatest contribution of an Indian scientist. Then this is all about the primary structure. Then the next one is secondary structure of the protein. The protein secondary structure is a three-dimensional form of the local conformation of the protein. This is the most important structural part of the protein in order to perform the function. Okay, the two most common secondary structure elements are alpha helices and beta sheet, and some super secondary structure called beta turns and omega loops. These are super secondary, these are regular secondary structures. Okay, the secondary structure elements typically form as an intermediate before protein fold into its three-dimensional tertiary structure. Okay, so this will form a particular protein fold, and that will give you the three-dimensional structure. The secondary structure is defined by the pattern of the hydrogen bonds between the amino hydrogen and the carboxyl oxygen. Amino hydrogen and carboxyl oxygen in a polypeptide chain. We will see how it is produced. It can be defined based on the regular pattern of the backbone dihedral angle. Okay, like Ramachandran plot says, okay, the regular pattern of backbone dihedral angle in a particular region of Ramachandran plot. Okay, I told you the secondary structure will produce only the psi and phi angle if it's fall in a particular region, not all the regions. Okay, so that is very very important. Particular region of Ramachandran plot, regardless whether it has correct hydrogen bonds or not. Okay, this is even though the hydrogen bonds are correct, it should have this particular psi and phi angle values. And the secondary structure of the proteins can be classified into two groups: regular secondary structure and irregular secondary structures. Irregular secondary structures are known as super secondary structure. Regular secondary structures are helices and sheet. The helices are alpha helices. Okay, there are two types depending upon the screw signs: right-handed alpha helices and left-handed alpha helices. Then 310 helices, then pi helices. Then the sheet are beta sheet. There are two types: parallel beta sheet and anti-parallel beta sheet. So what is the major important? The major force that stabilizes by the secondary structures are peptide bond and hydrogen bond. The hydrogen bonds are a weak interaction that stabilized by the peptide bond. Okay, so I told you this particular primary structure, due to the hydrogen bonds, if it is folded in the form of a zigzag manner or an asbestos sheet, that is known as a sheet, okay, or it form a helical like structure due to hydrogen bonding, then it produces a secondary structure. Okay, that is what it is very very important. So the most important thing is a peptide bond and hydrogen bond. And there is a, this is the alpha helical structure, and this is the particular protein that has got to both the alpha helical structure and beta plated structure. This is the red, okay, it's known as a sheet, okay, and the yellow is known as the helices. Okay, and this particular structures are mainly studied by three important scientists: Carl Linus Pauling, Robert Corey, and Hermann Branson. Okay, so these three scientists, they were identified most of the helices and sheets. Okay, and Carl Linus Pauling is the great person. He contributed. He is the person he studied the conformation of the nitrogenous bases, as we have studied in the previous unit. Okay, he studied about both the nucleic acid and protein. And for this particular study, especially that structure of collagen and structure of keratin, all those structures they have identified. Okay, for their great contribution, they have identified they have recognized for the Nobel Prize, and they got the prestigious Nobel Prize. That is what I told you. The secondary structural studies also won the prestigious Nobel Prize. These three scientists won the prestigious Nobel Prize: Carl Linus Pauling, Robert Corey, and Hermann Branson. Okay, so this is the historical milestone. Now we are going to study what do you mean by alpha helical structure. The alpha helical structure, simply called as alpha helix, is a common motif, very very important motif. Okay, this is a one important motif. What are motifs? As I said, it is a small structures of amino acid that has got a particular structure and function. Okay, then the small structures only forms a particular helix or a sheet. Okay, so that is known as alpha helical common motif. In the secondary structure of the protein, in which a right-handed helical conformation, in which every backbone, the amino group hydrogen bond to the backbone CO group of the amino acid located 3 or 4 residue earlier along with the protein chain. Okay, the amino terminal end will be of one amino acid, will be folded and interacted with the fourth amino acid, fourth carboxylic acid group of the same polypeptide chain, and form a helical like structure that is known as a alpha helix. And this particular structure, I told you, this is developed by these three scientists, identified by these three scientists, hence it is known as Pauling, Corey, Branson helix. Pauling, Corey, Branson alpha helix. So the amino acid in an alpha helical structure is arranged in a right-handed helical like structure, where each amino acid residues corresponds to 100 turn in the helix. That is, the helix has 3.6 residues per turn. Okay, one particular helix has the turn has 3.6 residues. Okay, and the translation of 1.5 angstrom or 0.15 nanometer in the helical axis. The pitch of the helix, that means to say the vertical distance between consecutive turns. Okay, the pitch, the vertical distance between consecutive turns. Okay, that is 5.4 angstrom or 0.54 nanometer, which is the product of 1.5 into 3.6. This 1.5, if you multiply with this 3.6, you will be getting this. And the amino group of an amino acid form hydrogen bond with the carboxyl group of the amino acid in the fourth residue. That is what I told you. The amino group of first amino acid will be interacted with the carboxy terminal group of the fourth amino acid. That means to say I plus 4, that is I hydrogen bonding. Okay, I plus 4 I hydrogen bonding. That is the most prominent characteristic of alpha helical structure. Then if it is 3-turn helix, I plus 3, that is I hydrogen bonding. Okay, instead of the first residue, instead of the fourth residue, this is the third residue will be produced in the case of 3-turn helices. 3 residues will be there. The next one, pi helixes I plus 5. Okay, it is interacted with the fifth, the amino terminal end of the one amino acid will be interacted with the fifth, the carboxy terminal group of the fifth amino acid. That is known as pi helix. Okay, generally this alpha helical is a tightly packed and no free space within the helix. Okay, this is tightly packed and there is no free space in the helix. Okay, then the amino acid side chains are often on the outside of the helix and point roughly downwards. Okay, it is point roughly downward. So these are the characteristic features of alpha helical structure. So this is what I told you. Here you can see CNH2. Here you can see it is going like this. Okay, this is going like this. That's what I told you. Here the R1, R2, R3 and R4. Here you can see the first NH of the first amino acid will be interacted with the CO of the fourth amino acid. Due to this, there is a helical like fraction is happening. Then the second will be with the fifth, third will be with the sixth, and so on. Here you can see N, R. Okay, this is the first R, second R, third R, fourth R. Okay, the first R has an OH group and the fourth R goes on an NH2 group, and there is a formation of peptide linkage. Then you can see in the second and fifth. Okay, the second one has got an O group and the fifth one got an NH group, and you can see the peptide bond. This is what I told you, 3.6 residue turn. Okay, this is carboxy terminal, this is amino terminal. This is simplest diagram, this is the detailed diagram. Okay, so this is known as alpha helical structure. Then there are two types of alpha helical structure. If it is oriented to the like this, the left-handed alpha helical structure. Okay, the screw will be in the left-hand direction. Okay, then if in the right-hand direction, this is the common thing, right-handed helixes, that is known as okay, this is in the right-hand direction, that is known as right-handed helix. Okay, this is what known as the amino terminal and carboxy terminal length. Okay, so I have already told you. So there are two types, left-handed and right-handed. Left-handed means it will be like this, right-handed means it will be like this. Okay, and the conformation is the same thing, and typically it will be right-handed helix. Left-handed helix is very rare. So this is all about alpha helical structure. Then beta sheet structure. Then the next important types of structure is known as beta pleated structure. Simply call it as beta sheet. Okay, there is a pleated. Okay, so it's another common motif, a small stretches of highly conserved amino acid are known as motifs of a regular secondary structure of the protein. The beta sheets consisting of beta strand connected by at least 2 or 3 backbone hydrogen bonds, forming a generally twisted pleated sheet. That is known as twisted, that is known as pleated. Okay, so a beta sheet strand is a structure of polypeptide chain, typically 3 to 10 amino acid long, with a backbone in an expected conformation. It's an extended conformation. So this is first studied in 1930 by William Osprey. Like DNA, he is the first person to conduct the extracrystallographic studies, and he studied the beta sheet. Then later, a refined structure was proposed by Linus Pauling and Robert Corey. That is what I told you. Both primary, the helices and sheets are proposed by Linus Pauling and Robert Corey. So the majority of the beta strands are arranged adjacent to other strands to form an extensive hydrogen bond network with their neighbors, in which the amino groups in the backbone of one strand established the hydrogen bond with the CO group in the backbone of the adjacent strand. I know that you did not understand. We will see this one. The main difference in the case of a alpha helical structure, only one polypeptide chain is there. The same polypeptide chain, first carboxy terminal end will be interacted with the fourth amino group that has got the the fourth amino acid that has got an amino group. Second with the fifth, third with the sixth, and so on. The same polypeptide. Okay, but here it is different polypeptide. One strand, next is the next strand. So this is what I told. This is known as a beta sheet. Okay, here you can see one strand CNHCOCHR NH. Okay, one, then CNCONH peptide bond. Second amino acid, third amino acid, fourth amino acid. Here you can see this is going like this. It is this is one particular strand. This is the next particular strand, and these two strands are connected by peptide bond. Okay, and this is known as pleating. This is known as a pleated structure. So the amino terminal end of one amino acid of one chain will be interacted with the carboxy terminal end of the next adjacent amino acid that has got an adjacent chain. Okay, here it is NH, it is CO, this is CO, then it is NH. Opposite direction you can see. This is NH, this is CO. Okay, here you can see R1, R2, R3, and like this you can see the bond. It's very simple, and this particular structure is known as beta plated structure. This is the another important. Here you can see 1, 2, 3, 4, 5, 6, 7, 8 plates. Okay, like that you can see that particular plate as zigzag thing. Okay, this is hydrogen bond. So due to the hydrogen bond only the secondary structure is producing. And there are two types of beta sheet. One is known as anti-parallel beta sheet. Next one is parallel beta sheet. As the name indicate, anti-parallel means, here you can see, first we will discuss parallel. Okay, here you can see the parallel C alpha CONH, C alpha CONH, C alpha CONH. Like this is amino terminal end, this is carboxy terminal end. The amino acid is orienting this direction. Next amino acid chain is orienting to this direction. Next amino acid chain is orienting to this direction. Next one will be this direction. So what is that? Here you can see parallelly it is going on. Okay, parallelly, this is same level, parallelly it is going on. Then what is happening? In this particular case you can see N and H, and here you can see this is what I told you, the NH of one amino acid of one chain, one strand will be interacted with the carboxyl group of the another amino acid, that is the next chain, next strand. There is a formation of a OH, that is hydrogen bond. Here you can see CO, then you can see like this, then you can see NH, then here is this CO. This is NH, this is CO, there is a bond. This is NH, this is CO, there is a bond. Okay, this is CO, this is N, there is a bond, and so on. Throughout you can see it's highly stabilized by the network. That is what I told you, extensive network. Okay, this is known as parallel beta sheet. Then what do you mean by anti-parallel? So here one particular direction chain will be N C alpha C double bond O, N C alpha C double bond O NH, C alpha C double bond O NH. Like this it will go, and the same thing will go from it is coming from top to bottom. From here it is going towards bottom to top. Okay, here you can C alpha C double bond O NH, C alpha C double bond O NH, C alpha C double bond O NH, and it will go like that. One is oriented this direction, another is oriented to another direction. Here you can see in that particular case, this one also, one is in this direction, another is in another direction. Okay, and here you can see this is NH, the amino group of one amino acid in the one strand will be interacted with the carboxylic acid end of another amino acid that is present in the next strand. There is a hydroxyl group. Okay, this is also C double bond O. Here you can see C double bond O of one amino acid end and NH of another amino acid of the next strand. Okay, here you can see hydrogen bond. Okay, this two hydrogen bond, two hydrogen bond, two hydrogen bond. Okay, very nicely. The fashion is very particular conformation. Okay, and parallel it will be like this. Okay, anti-parallel it will be like this. Okay, so anti-parallel will be more stable than parallel. Okay, so because of steric clashes. Okay, so this is very important. So this is all about the secondary structure of the protein, dear students. So this is one of the most important topic in this particular unit. So now we can conclude and we can have a glance of this particular class once again. We have already discussed what is a protein structure. We have discussed introduction. We have studied what is a polypeptide. Okay, what are the major interaction, what are the conventional experimental approach to study. Why do you want to study structures? In order to study the function. What do you mean by peptide bond? What are the bonds? Okay, how will it will produce the formation of peptide bond? The amino terminal end of one amino acid will be interacted with the carboxyl terminal end of the next amino acid. Then the salient features of peptide bond. What are the important salient features? Especially the torsion angle. Okay, psi, phi and omega. Psi and phi are rotatable and omega is non-rotatable. And what are the major torsion angle in the peptide bond? Then we have discussed, based on all these basics, protein structural configuration: primary, secondary, and super secondary, tertiary and quaternary. In this particular class we have studied primary structure of the protein in detail, the structure of primary structure of bovine insulin by Frederick Sanger. Okay, his contribution, Ramachandran plot, the contribution of Indian scientist from Indian Institute of Science. Okay, Govind G.N. Ramachandran. Then what do you mean by Ramachandran plot? This is very important. Then that is based on the psi and phi angle of the polypeptide chain. Then secondary structure of the protein. What do you mean by secondary structure? It's a local conformation of the protein. That is known as secondary structure. Types of secondary structure. There are so many types: alpha helices and helices and sheet. Helices are different types. Alpha helices, right-handed helices, left-handed helices, three-handed helices, pi helices, etc. Then sheets are parallel and anti-parallel. What are the historical milestone? Okay, especially Robert Corey, Linus Pauling and Hermann Brandtmann's contribution. Then we have discussed alpha helices in detail, types of alpha helical parallel, right-handed alpha helices and left-handed. What do you mean by right-handed and what do you mean by left-handed? Then next one, beta sheet. What do you mean by beta sheet? What are the characteristic feature? How it will produce? What are the different types of beta sheet? Okay, parallel beta sheets and anti-parallel beta sheets. How their conformations are producing. Dear students, please draw the diagrams and try to learn the structure. This is very important. Without drawing the structure, without drawing the chemical illustration, you will not get the mark for this particular chapter. Please note that. Okay, you have to study that thing carefully. So these are the textbook, Principles of Biochemistry by Leninger. All the textbook you will be able to see this particular information. Okay, so thank you, dear students, for watching this particular class. If you are having any clarification, you can ask me. Thank you very much.